Hello, and thank you for joining us. Today's webinar is Prescriptive and Intelligent Sample Dilution for Online ICP OES Analysis. Our presenter is Pete Winship, Technical Product Manager for Teledyne CETAC. If you have any questions during the presentation, please submit them throughout using the question function, and Pete will answer them individually offline. This webinar is being recorded and will be available on our website within a couple of days. Okay, now I'm going to hand it over to our presenter, Pete Winship.
Lovely. Thank you very much for the kind introduction. Hello, everybody, and thank you for joining us. So as Sally noted, my name is Pete Winship. I'm a product manager at Teledyne CETAC Technologies. One of the systems that I'm responsible for is the SDX system, which I'll be discussing today. But today I'm going to describe prescriptive and intelligent sample dilution and how that is applied to ICP-based analysis using this technology. So just to start, I just wanted to sort of set out a quick agenda just to walk through what I'm going to be describing today. So firstly, I just wanted to discuss what we call the analytical workflow. So this is a workflow that can be applied to a lot of different types of analytical technology. In this case, I'm going to be describing it with respect to ICP OES. This is a workflow that I've described on previous webinars. But I'll be going through this as the starting point, the jumping off point for the rest of the webinar. From this workflow, I'm going to be focusing specifically on dilution processes for preparation of samples and calibration standards, and then also repeat analysis and how the SDX system can meet those requirements. I'm going to describe the SDX system and what it is, what it can do, introduce that and go through a hardware tour. I will go through briefly on compatibility with ICP OES instrumentation, and then from there I want to essentially sort of theoretically apply what we've been describing through workflow, through the SDX system to certain applications, and again with respect to dilution processes and repeat analysis. So that's the agenda for the webinar, so I shall make a start. So the analytical workflow. So as I mentioned, an analytical workflow can pretty much be applied to any kind of analytical technique. There will be some deviations, but there are a lot of similarities to this. So I'm going to start off with what we all have, which is we all start off with a sample, which is represented here in red on the left-hand side of the screen. And what we want to get to is data, which is represented in green on the right-hand side of the screen. That data needs to be representative of what is in the sample. So analytes that we're interested in, the data that's output, what we want is we want that representation so we can report that data and possibly act on it. So in terms of ICP OES analysis in this workflow, what comes after we have a sample? Well, the next thing in this case is preparation processes. So what we might do is this sample might be in a solid state, a solid form, it might be in a liquid form. So to start off with, if it's in a solid form, we need to get it into the state where it can be introduced to an ICP OES instrument, which is typically via a glassware arrangement, nebulizer and spray chamber. So if we have a solid sample, we need to work through some sort of breakdown process, a digestion process. The result of that digestion process might be a very high acid concentration solution that we then need to dilute from there so that we can work with it with ICP OES. So following a digestion, if it was required, we might need to undertake a dilution. If a sample is in a liquid form, then we might just go straight to the dilution process. So via digestion and dilution, or just dilution, we take our sample and we get it into a state where we can introduce it to an ICP OES instrument. Now, what we would most likely be interested in is quantifying the analytes of interest in this sample. And in ICP OES, we're interested in quantifying elements, so elements of the periodic table. So what we would need to do to quantify the analytes of interest in that sample, we need to prepare calibration standards to compare it to. So we take our signal data from our unknown sample, compare it to the signal data from calibration standards, and that way we can quantify the analyte of interest. Now, as part of that quantification process, there will be certain quality aspects to it as well. So we would need blank solutions as well. So we know what our background level is, our baseline levels from that measurement. And then also we might need to prepare some quality control standards for quality assurance of our analysis generally. So that's where we would start. Once all of this is prepared, we would then be looking at the next step, which would be introducing this to some sort of instrumentation. So in this case, ICP OES instrumentation. So that introduction might be a manual process, and we might introduce these manually. It might be automated, which these days is more often than not. So we might need some form of automation to introduce our samples, calibration standards, QC samples, blanks, etc., into our instrumentation for measurement. After we've measured the analyte content and established our signal data from our samples, we then have some form of data output. So in the case of a lot of instrumentation that's commercially available in the market today, this data might have been processed by the operating software, and what we actually get as an output is our end data point. It might not be. We might end up with raw data that needs processing, so the analyst, the operator might need to take that data, process it in some sort of software package like Excel or similar, and then arrive at the final data. So we have data out of this analytical process. If everything has progressed as we want it to, that will be the end point, but it might not be. So once we've generated our data, it essentially needs assessing, and we need to start asking some questions here. So first of all, we might be asking these kinds of questions. So our calibration standards, from our calibration standards, we prepare calibration lines. Is the quality of those calibration lines, are they good enough for quantification processes? Is the R² value on that calibration line acceptable? Is it within the tolerances that we have established as part of our analytical design? That's the first question to ask. Are we using internal standards as part of this to account for any kind of sample matrix signal suppression or enhancement? Is the internal standard signal of our samples, is it within the tolerance again that we set out in our analytical design? The answer might be yes, or it might be no. And then our QCs. Have our QC samples, do we have agreement of the measured analyte concentration against the known analyte concentration that we have from information we have on these QC samples? Is there agreement between the two? If we have a batch analysis, which is quite a large batch, and we might have 10 QCs, is there a criteria for pass or fail here? You might have a criteria where you have, if you have 8 QCs that pass out of 10, that might be an acceptable batch analysis. So these are questions that we ask. Now, if the answer is to these questions, are our calibrations acceptable? If the answer is yes. If our internal standards are within tolerance, if the answer is yes. And if our QCs have passed, if all of the answers to those are yes, then essentially that's our endpoint and we have our data. But if the answer is no, and it's not acceptable, then we might need to look at a reanalysis process, which takes us to the next step. Now, that reanalysis, we might have sample solutions still remaining to repeat the analysis, but it might take us back to the same point. It might essentially confirm what we've done before. We get agreement with what we had before. With, again, our calibration is not what we want, internal standards might be out, our QCs might have failed. So we might have to go right back to the start and reprocess everything so that we can carry on. So that is our workflow that I want to sort of work through today, but I'm going to focus on two areas. So the first area I want to focus on is this one. So with the exception of digestion, I'd want to focus on dilution processes, calibration standards, etc., and what is involved in undertaking that. So let's start with our calibration standards. So we will have for our analytical batch criteria for calibration standards. We will have an idea of a calibration range that we want to work within, and we will also have a number of calibration standards in mind that need to be prepared for our analysis. So I'm just going to start off, just show you this. This is a calibration graph, just comparing some data, analyte concentration in standards versus analyte signal, and for the vast majority, if not all of you, it will all look very familiar. So the calibration points are in blue there. And then we've got blanks and QC samples. We might have, so we'll have our blank obviously at the bottom of the calibration line. Then we might have a low QC standard and a high QC standard. So as part of our quality assurance. And then if all has gone well, we will have samples that need to be prepared, possibly diluted, and what we want there is we want these unknown samples to fall within the extremes of our calibration range. So just in terms of the dilution processes that we need to undertake for an analytical batch, we know that these things are, preparation of this is relatively time consuming. Depending on the size of the batch, that is. So if we have a full analytical batch where you've got a full auto sampler, if we've got a large number of samples to work with, then essentially the requirement for our calibration standards, our blanks and QCs, sort of multiply up with it. So the more samples that we have, the more QCs that we would need, the more blank solutions that we would need. And we might have multiples of the calibration standards. You might be sampling from the same tubes for calibration standards. You might have prepared them separately. Either way, there is a time that's required for preparation, dilution of these solutions. Typically, what you're doing here is repeat processes with pipetting. So pipetting small aliquots of certain liquids, and then dilution with dilution media with larger volumes. But if somebody is to be sat at a lab bench or a fume cupboard, a fume hood, then there is accuracy and precision questions or error associated with repeat analyses, repeat processes, sorry. And with the best will in the world, there is an accepted error associated with this. And then you've got essentially the cost of all of the preparation time. That might be financial cost and effort. It's essentially staff time and effort. So if that could be applied, that staff time applied elsewhere, that might be better for your laboratory processes and workflow. So that's the dilution side of things. Now, what happens if everything doesn't go well? So we go back to our workflow, and I wanted to focus next on this question here. So short of QC failure to a degree, what happens if we have a calibration line which doesn't meet our requirements, or our squares aren't particularly good? What happens if our samples exceed the analyte elements concentrations? What if they exceed the calibration range? What if our internal standards have fallen outside of tolerance? What can we do about that? If you've manually prepared everything, then again, essentially you're talking about manually re-preparing everything. That might be the entire batch. It might be a small number of samples, but it's never really a small number of samples, because to reanalyze samples, you also need to re-prepare your calibration standards, blanks, QCs, etc. So the first situation I just wanted to suggest was this one. Unacceptable samples. So samples with analyte concentrations that might exceed the top standard. Now, it's in some processes, some analyses, it might be acceptable to extrapolate your calibration line so that it meets and incorporates your unknown samples that have exceeded the top standard. That might be acceptable. That it depends entirely on the analytical design, the analysis that you're undertaking. It might not be acceptable at all. So it might be, there might be certain criteria, and there's one for a water method, which I'll mention later, where that's not acceptable at all. So also, our internal standards, tolerance range that we want to work within. If we have a scenario where our sample matrix suppresses our internal standard signal, then it will be doing the same for the analytes of interest to us in our unknown samples. So that brings in another area of error. So if you've got a situation like that, then a repeat analysis is probably likely. Pipetting errors, I've already mentioned, accuracy and precision, R squared of our calibration. Do our QCs agree? It could be that QC disagreement could come down to pipetting error. And then, as I've mentioned as well, reanalysis of samples, even if it's just a handful of samples from your analytical batch, is not just the analysis of those samples, it's another set of calibration standards, blanks, and QCs that require preparation as well. So that's where I want to leave that part of the discussion for a minute. The workflow and these two areas where we can use automation to improve this process. So to improve essentially the labor of undertaking sample dilutions, and also the issue of reanalysis. So I'll leave that there, and then the next step we need to do is to introduce to you the SDX system, and then show you how this can factor into that workflow and address these issues. So first of all, what is this system and what can it do? So the SDX system, this is an online intelligent automation system for the dilution of samples, calibration standards, etc. for ICP-based instrumentation analysis of aqueous-based, acid-based samples. So that's what it is. What can it do? Well, this system can offer 3 different types of dilution process. Well, 2 really, because the first one is no dilution. So we can use this system and the central automation to primarily, with no dilution, use it as a pass-through. So uptake our samples and introduce them to an ICP OES system. So we might not need a dilution process. Then we can use a prescriptive dilution process. And that process can be for our unknown samples, and we can apply various dilution factors between 2-fold and 5,000-fold for this process. And then we can introduce them after they've been diluted to our ICP system. We can apply that prescriptive dilution and those specific dilution factors as well to our calibration standards, all of which will need a different level of dilution from a multi-element stock. And then we have the third option, which is our responsive dilution. So I'll provide you with more information on this as we go further along. But the responsive dilution, the dilution factor associated with that, is variable depending on what's required for that sample as and when it's failed some criteria in the batch analysis. And then this system as well, it is compatible with our AS Express Plus rapid sample introduction system, so that after we've diluted our samples and they're ready for introduction, we can accelerate that process with the AS Express Plus system for sample introduction and analysis. So this system essentially it takes up aliquots of or multi-absolute that we want to dilute. So it will accurately take up a sample aliquot, a standard aliquot, and then it will also at the same time take up a volume of diluting media, and it will bring the two together for mixing. So, and this will do this in either a single dilution process, or we can do a serial dilution process. And the two are brought together and they're mixed in a vortex mixing vessel. So we ensure that we've got a homogeneous sample ready for introduction. And then as part of all of this, and I'll show you this later on, we've got configurable washout processes with this system as well. So for applications where you might have samples that contain a very high analyte concentration, or you've got samples that contain analytes that have known memory effects, for example, something like iodine or mercury, for example, we can configure our rinse processes, our rinse cycles, washout cycles, to make sure that after we've done a sample introduction, we're ready for the next sample, and there'll be no carryover of analyte signal from one sample to the next. So bringing in that accuracy and precision that we're interested in. So just back to the dilution modes. So firstly, a no dilution mode. So we can select this as an option for any and all of our samples in our batch list if we were so inclined. So essentially the system operates as just an auto sampler. So we are introducing samples from our automation sample racks to our ICP OES. This mode could be used for our blank solutions. So if we've already prepared our blanks in this case, we can just introduce them from the automation. It can also be used for samples which are already diluted or might not require a dilution step. So water samples in a water lab, that's a good example of that. So that's the first mode. Now, the prescriptive dilution, so what we can do is we can basically establish a dilution factor within the range of 2-fold to 5,000-fold, and for everything in our batch list, again, if we were so inclined. So we can establish a dilution factor for each sample, each of our calibration standards, our QCs, our blank, if that's how we wanted to approach it. And those dilution factors are defined by the operator, by the analyst. So based on this, we can use this system to prepare a full range of calibration standards from a single multi-element stock solution. So we put that multi-element stock solution, we might fill a 50 ml sample tube with that, put it in a standard position on the system, and then our system can then introduce an aliquot of that multi-element standard, dilute it, and then we introduce it to our ICP system and get our data for any and all particular calibration standards. So we can prepare a full range. We can then, for our unknown samples, if it's a relatively straightforward analytical process, excuse me, we can use a blanket dilution factor for all of our samples. So for example, something like a 50-fold dilution factor for all of our samples, if that would be appropriate. And then, but instead of that, if it's a different kind of analysis, we might need a mixture of dilution factors for different samples on our as part of our batch. Some of our samples might need a 50-fold dilution, others might need a 200-fold dilution. But you can apply these different dilution factors to each individual sample if you wanted to. And then the third mode, so the responsive dilution. I've already mentioned a fair bit about this, but if we have a situation where we have a sample which exhibits an analyte concentration which exceeds the top standard, this sample is recognized, and then based on the signal that's exhibited by the top calibration standard and the signal that's exhibited by this unknown sample that's been recognized, then a new dilution factor can be recalculated to bring that sample down so that it falls under the top standard. Now, typically we do this to about 60% of the top standard signal, but we can vary that. Once this sample's been recognized, it will undertake this process of redilution and reanalysis, and it can do that there and then, so that that sample is reanalyzed within the same QC criteria as the first analysis of that sample. So we're not losing anything in terms of our QC process. So if you have a QC every 20 samples for the sake of argument, then that sample can fall within that QC criteria. And then similarly, for internal standard suppression, if we have samples whereby the sample matrix causes us a suppression problem of our internal standard or our analytes of interest or analyte signals of interest, then those samples are recognized. And if, for example, the tolerance on our internal standard is plus minus 10%, then we can do, the system will perform iterative redilution of that sample until the tolerance falls back within that, that plus minus 10% again. So that's the third mode of dilution. Now those three modes can then be applied to whatever the scenario is with our preparation processes, so the prescriptive dilution, and then the responsive process, the responsive dilution, can be applied to any reanalysis that we need as part of the process. So, in a nutshell, that's what the SDX system does. Now the hardware. So I'll just walk you through the hardware, and then I've got some video footage to show you on how the system works altogether. So firstly, our ASX 560 autosampler. This is the central automation of this SDX system. So this is from the place from which our samples are taken. So this is our robust industry standard. It can accept up to 4 sample racks of samples of various sample tube sizes. The system itself is manufactured from molded polymer and carbon fiber components, so it's robust to various laboratory environments for trace element analysis work. There's no exposed metal, which obviously you don't want in trace element work. And then it's got a high resolution XYZ movement, and that XYZ movement is configurable, so we can speed it up or slow it down depending on how we want to operate it. So added to that, what we then do is we add some additional components. So instead of the, for the 560 system, we add a new sample base tray. So what you'll see here is that that is the same as the standard base tray for the 560 with one exception. Now there's no standard positions at the back. So that's the base tray we use for the SDX. So what we do then is we add the next component, which is this vortex mixing module and standard positions assembly. So what you'll see on the left side of that component, you'll see there the vortex mixing vessel. And then to the right of that, you'll see a new set of standard positions. So these are reorganized so that everything fits together in the place where the standard positions used to be on the 560. And then added to that, we've got a syringe pump module. Now this syringe pump module is responsible for our sample aliquoting of and take up of the diluting media that we need, and then ultimately the dispensing of sample media into the vortex vessel. So what you do is you put all of this together, and there's a couple of versions of this. So first of all, you'll see the setup on the arrangement on the top image there. That's what we refer to as our standard system, standard SDX system. So the 560, the ASX 560 automation with the SDX components with it. Now, in terms of bench space, what you're looking at there is how the system could be set up. So in terms of bench space, you are adding the syringe pump module to the footprint of the ASX 560. You could also, and we have this as part of our standard system, we can mount the syringe pump module to the ASX 560, so it just sits above and a little bit behind the vortex vessel and rinse station positions, so that the only footprint that is taken up is the standard ASX 560 footprint. And then if you want to add the AS Express Plus system to this, then the only footprint that you're adding there is the vacuum pump module and the electronics module as well, which typically sit next to your ICP OES nebulizer. So in terms of footprint, hopefully you'll be able to see this in process. I've got some footage here of the SDX system in operation, so I'll just walk you through this. So hopefully you can see this. So this is our SDX system. So our SDX syringe pump module is part of this system. Now next you're going to see our ASX560 automation with the SDX components in our laboratory in Omaha in the US. And we'll just walk you through a standard sample introduction to an ICP. So we have our vortex mixing vessel in standard positions at the back of the automation, and the syringe pump module there to the left of it. And we have a sample here, which is just a blue dye. And we'll start off with just a dunk of a sample probe into the rinse station. We have a priming process just to make sure the lines are clear. And then we take an air gap and an aliquot of sample up into the sample probe, and that's under the action of the syringe pump. Next, the sample probe takes a dunk in the rinse station, and the syringe pump module takes up the aliquot of mixed and diluting media. And then all of that is dispensed into the vortex vessel where it's mixed and homogenized and ready for introduction. The sample probe goes to the rinse station for a quick rinse, and then the system treats the vortex vessel like a sample tube, and that sample is carried away via a peristaltic pump or by our AS Express Plus system to an ICP OES instrument. As you can see there. So next, if there's any excess sample remaining, that is drained out of the bottom of the vortex vessel, and we introduce some cleaning media, some cleaning reagents into the bottom of that vortex vessel, where it spins again and cleans the vessel, and then that cleaning agent is drained out of the bottom. So that's one of the things that we configure to make sure that everything's ready for the next sample, and then we move on. So that's how the system operates. So from here, I'll briefly talk about compatibility, and then I want to talk about some application examples. So compatibility, we currently have the SDX is currently compatible with two different types of ICP operating software. So that would be Thermo's Qtegra for ICP OES and ICP MS analysis. And then we've got integration as well with Spectro's ICP Analyzer Pro software for ICP optical instruments. And then later in the year, we've got other ICP OES instruments which are going to become compatible as well, possibly with a third. So how does the system work in terms of compatibility? So for the thermo instrumentation, we have prescriptive dilution for standards and samples. The Qtegra system will recognize when a sample is outside of calibration range. So we've got responsive dilution, we've got responsive dilution for a failed internal standard, a dilution factor up to 5000x. It can do serial dilution, and it's compatible with our AS Express Plus introduction system. For the spectro instrumentation, we have prescriptive dilution for samples, and we've got the ability to respond to samples that fall outside of calibration range. We've got a dilution factor up to 100x, and it's compatible with the AS Express Plus for operating the software versions beyond 35. And as I mentioned, there's other ICP systems coming online with the SDX later in the year. So examples. I've got two key examples that I want to work through with you, but I'm just going to start here with the idea of application. The SDX system can be applied very broadly, and it's applicable to many of the same applications that are typical of our ASX 500 series automation. So that could be applicable to typically aqueous acid-based samples. It's certainly applicable to environmental samples in environmental laboratories, so soils digests, waters, etc. It will meet the criteria for EPA 200.7 and EPA 200.8. And certainly for laboratories outside of the US that don't work with the EPA methods, it will meet the criteria, the quality criteria and methodology laid out for waters and soils, etc. Mining samples, this is something we've got expertise in as well. So mining extracts which have been digested in high concentration and acidic media, we can work with those as well. Essentially any kind of general digest testing we can work with. And then things like foodstuffs, crops analysis, etc. And anything outside of that whereby we've got liquid dilution, which is a requirement of the analysis. As we're looking at other applications all the time for the SDX system, and also its sister product, the SimPrep, and we're particularly looking at the moment at more viscous sample types, viscous liquids that we might need to work with for dilution steps. So one of the obvious applications that we're looking at the moment is things like bloods analysis and blood plasma analysis, where you've got essentially a liquid sample which is more viscous than samples that you might be working with in terms of digests and waters, etc. So that's something that we're looking at, and application notes are being prepared for that, and they'll be made available to another time. So from that list, what I wanted to focus on was two areas of application from that list. So I'm going to focus on foodstuffs and crops, etc., and then also waters analysis. But as part of this, part of the discussion, what I wanted to do was sort of bring in and really focus more on the dilution required and the responsive dilution if there is an analytical failure as part of the batch analysis. So starting with foodstuffs and crops, we require data, certainly elemental data, on foodstuffs and crops from essentially 2 points of view. So you might be interested in elemental composition of foodstuffs, etc., on a nutritional basis. So you might be interested in analytes such as selenium or copper, zinc, that kind of element, whereby there might be a requirement, a quality requirement for certain nutritional composition. You might also be interested in element composition from a contaminant point of view, so from a heavy metal composition point of view. So, for example, elements like arsenic or lead or cadmium. I certainly know that there's been focus in recent years on the arsenic content of rice, but that's the other side of the coin in terms of the data required for this kind of analysis. You might also, I mean, for foodstuffs, there's also quite a mix of levels of concentration for analytes you might be interested in. So for some analytes, selenium is a good example of this. You might be looking at low concentrations, so you might be looking at the ppb level of concentration in terms of sample composition. And then for elements such as calcium, you might be looking at much higher levels, so the ppm level of concentration. So it's going to be analyte dependent, it's going to be sample dependent. For this kind of analysis, like most kinds of analysis, you might be looking at this from a quality control point of view. So you might have a good understanding already of the kind of level of concentration of analytes in your sample, and that from a quality control point of view, you might be looking at just confirming whether or not you are above or below by a certain magnitude of concentration for your analytes. So in that scenario, you might have a nicely established calibration range to work with. You might not be expecting to do any kind of reanalysis after the process, after the analytical process, but you would still need to undertake your preparation processes. So the SDX would come into that kind of analysis, certainly. Your samples might be completely unknown. You might have such a mix of samples, so many of your analytes of interest might fall quite nicely within your calibration range. They might not. Some of your analytes might far exceed what you're expecting, hence completely unknown. So for foodstuffs and crops, typically, because you're working with a solid sample, you're going to need to do some form of digestion or breakdown on your sample to get it into a liquid state. And that's typically going to be undertaken with an acid-based media. And then you'll need to do a dilution to volume beyond that to reduce the concentration of your acid, digest acid that you've used. Bring it up to volume and you can work with it from there. So in this case, in the case of the application for foodstuffs, crops, etc., the SDX can be used for your standard preparation, your sample preparation. It might require it for sample dilution. Certainly from a preparation point of view, you may then need to use it for a responsive point of view afterwards. And then obviously your sample introduction. So in terms of calibration preparation, we've talked about this already, but one thing I just wanted to point out was this is some data for a series of analyses that we did on food testing, so food digests. The SDX was used to prepare our calibration standards, and these are some R-squareds that we got from our preparation of calibration standards for this. So we've diluted our calibration standards from a single multi-element stock solution on the SDX system, prepared them all in the vortex vessel, introduced them to our ICP OES, and the diluting media was 1% nitric acid, and we've arrived at our nice calibration lines with a really good R-squared value. Now what do we get from that? So against our calibration standards, we've run a series of samples. So in this case, certified reference material, bovine liver, rice flour, and milk powder. So we've digested all of these and we've diluted them from there and then analyzed them against our calibration standards. So what I wanted to show you here was I mean, first of all, that we've got good recoveries of these certified reference materials for the measurements against what they should have been. But what I wanted to focus on here was not that so much, but more that we've got two types of data here. So we've got our good recoveries, but of this data we're looking at, we've got, first of all, analytes that are being measured at the ppm level, so much higher concentrations, and that's the top 5 analytes, and then the lower 7 analytes have been measured at the ppb level. For all of these elements, we've got two types of data. One is where the data that's highlighted in green, where we've introduced these samples and the measurements have been undertaken straight away. The analytes have fallen within our calibration range. We're very happy with that, and we've collected the data. And then there's the data highlighted in yellow, where the elements of interest have required a redilution process. So the system has recognized that certain analytes fall outside of the calibration range, and a redilution has been triggered. And then we reanalyze after we've done a redilution, and then everything stays within the QC criteria of our batch analysis. So in this case, what we've done is we've analyzed a series of samples, many of which have elements which exceed our calibration range. So what we've done is the SDX has handled the redilution processes for certain samples. It's kept it all within the same batch, so there's been no requirement for a redilution after the fact, sorry, reanalysis, a full batch reanalysis after the fact, or a repeat of certain samples after the fact with new calibration standards, etc. So if, for example, this was set up to run overnight, the analyst would return the following morning and have all of the data that they would need without needing to do a reanalysis. So just to give you an example of this in terms of responsive dilution, so for the rice flour data, there were analyte signals for phosphorus and sulfur which exceeded the top calibration standard. So this prompted the redilution process. Now, based on the signal that was exhibited by the top calibration standard, and also the signal from the unknown sample, a redilution factor was calculated by the system of around about 4-fold dilution, and that would bring that sample down to within 60% of the top standard. And then we undertook our reanalysis and collected our data. So that's just an example of foods analysis. So essentially a digest analysis, in this case on foods, and how we use the SDX in terms of preparing calibration standards, diluting samples for sample introduction, and then how the system handles reanalysis of individual samples as and when required according to certain criteria, so that everything is done within the same batch. That batch has just been slightly lengthened by the addition of some reanalyzed samples that the laboratory analyst hasn't had to interact with. So moving on from foods to water analysis. Now, I did talk about water analysis on another webinar last week, but in this case, I want to focus more on the requirements of a high throughput water lab. So, and in this case, I'm using EPA 200.7 as an example, but this could apply to any and all water laboratories around the world, whether they work with the EPA method 200.7 or not. So feedback that we do have from water testing laboratories is that certainly for wastewater analysis, these labs report that up to 25% of an analytical batch for these samples requires a reanalysis after a batch is complete. So the result of that is obviously a loss of laboratory productivity. So that being the case, as I mentioned before, if 25% of a batch needs reanalyzing, it's not just those samples that we need reanalyzing, it's those samples plus relevant calibration standards, QCs, and blanks. So you go from X number of samples, and that is multiplied up according to the methodology that you have. Also, this is the method I mentioned earlier as well. So EPA 200.7, this requires all samples to be quantified at a level which does not exceed 90% of the top calibration standard signal. So this is not a method whereby if you've done your analysis and you've got samples that exceed the top standard, you can't then extrapolate the calibration line and accept those samples. So a reanalysis of some kind has to be undertaken. So what I wanted to do here was just sort of work through the SDX system and how we can basically take this and apply it to water analysis. We can use it to prepare our calibration standards, our blanks, QCs, and also we can use it to either dilute our unknown samples, or we can introduce our unknown samples with a no dilution mode introduction, and then use the responsive dilution mode to catch any samples that have gone through and then exceed that calibration. So the intelligent response can be applied to make sure that we make sure that all of our samples fall within the calibration range that we need them to. So just to give you a few more details on this, so in how this can be applied to waters analysis. So we did some work previously with Thames Water in the UK and Thermo Fisher Scientific in the UK, whereby we took 15 unknown wastewater samples, which were provided by Thames Water, and 2 AquaCheck QC samples. And we've analysed these with respect to calibration standards, and we've got internal standardization in there as well, via the SDX and Thermo ICP OES instrumentation. So we've taken these samples and run them. Now the SDX has prepared calibration standards, so just a short 4-point calibration line. It's prepared our QCs, and it's done this for 15 or 16 analyte elements that you can see across the top there of this table. Now, I didn't want to focus too much on this. So the calibration standards, QCs, and samples were all prepared. Now, of the 15 unknown samples, these unknown wastewaters, 10 of them, because we've not diluted at all, we've just used it as no dilution as a pass-through, 10 of them have required some form of redilution because they have analytes which have exceeded the top standard. So in this case, for each one of these samples, the responsive dilution has been triggered. So we've reanalyzed these samples at a new dilution factor within the same batch. So there's no requirement for a reanalysis, no re-preparation of calibration standards, etc. And we've brought all of this down into the calibration range that we need to work within. So this is the output data from this. I didn't want to focus so much actually on the actual concentration data of these individual elements. What I wanted to point out here was these dilution factors. So what we have here is just some examples of the dilution factors which have been calculated to bring these samples down to that 60% of the top standard level. So anywhere between a 2-fold dilution and around about 140-fold dilution to make sure that we're down in the range that we want to work with. So that, I just wanted to show you that as an example of real-world samples being used on the SDX system, and how essentially we can have a scenario where we, it was the title of my last webinar, which was Rack Up Your Samples and Leave. You can essentially take these wastewater samples, put them in sample racks, leave them on the system, and then the SDX can do all of the work for you. So just to finish off, my application conclusion, why invest in the SDX system? Well, essentially what I hope I've shown you through this webinar is that the SDX system enables you to undertake prescriptive and responsive dilution with your samples so that you can avoid, first of all, incorrect quantification. So, and then also it allows you to avoid the need to reanalyze whole batches or large parts of batches the following day. So essentially, you are saving yourself cost, time, effort, etc. So your laboratory workflow can be improved. Accuracy and precision of your analysis can be improved because you are using automation for preparation rather than you having somebody doing repetitive pipetting processes, and you're minimizing error as well. So hopefully I've walked you through that through the material that I've shown you today. So thank you everybody for attending today. If anybody has any particular questions, please feel free to email me those questions, and I can respond to those over the next coming days. And then thank you very much for attending and your time. Thank you.